Well, hello everyone. It's my pleasure to chair the next two talks, both in imaging data analysis. First speaker is Dr. Stephanie Ling. She obtained a master's in biochemistry from the University of Oxford, then a PhD in the Institute of Cancer Research. And she works now as Associate Principal Scientist of the Imaging and Data Analysis Analytics Group in AstraZeneca. And she will talk about some imaging data analysis. Thanks.
Great. So today I'm going to tell you a little bit about what I've done over my career. So I did my PhD at the Institute of Cancer Research as well. And I'm just going to take you through some of the things I did then and some of the roles I've had since at AstraZeneca. So throughout my career, the thing that I have been most interested in is using different analysis techniques to try and understand biological complexity. So within tissues and cells, you have complex multicellular tissues. So you have multiple different cell types that have different gene expressions, different post-translational modifications that feed into different signaling networks, both within cells and then between cells. How these different cells are located relative to each other affects the biology, and the subcellular location of different proteins affects their activity. So all of this together, not just at the protein level, but also endogenous metabolite flow and flux, all affects the outcome that you see in multicellular tissues. And so the thing that I've been most interested in is finding ways to unpick all this complex information. So before I joined AZ, I was in the cell communications group at the Institute of Cancer Research, particularly looking at the interrogation of tumor-stroma signaling in pancreatic ductal adenocarcinoma. And to do this, I looked at a combination of mass spec proteomics and high content immunofluorescence. So some background for PDAC, which I'm sure most of you already know, but it's one of the most lethal forms of cancer, has very poor drug delivery and drug resistance, and a very dense fibrotic stroma. And this can form more than 50% of the cells, one of which is the pancreatic stellate cells, and they produce this dense fibrotic stroma that has been correlated with poor patient prognosis. In my PhD, what we wanted to know is how are these stellate cells activated in PDAC? What is their contribution and the role of stellate cells within the tumor microenvironment? So just really quick, what we found is that these pancreatic stellate cells are activated by factors that are secreted from tumor cells upon KRAS expression. So we did this both in direct co-cultures, but also using conditioned media, including with inducible KRAS cell lines. So we could see that these factors were being secreted in a KRAS-dependent manner. So we then did some mass spec proteomics using SILAC to see which proteins were increased in expression upon KRAS expression. In particular, we looked at secretomics, so looking at just that conditioned media to find out what growth factors were secreted. We used things like gene ontology to pull out which were the cytokines and growth factors that were upregulated. We then were able to take each of these in and put them exogenously onto cells and see which were causing this activated phenotype. And we saw several that were causing things like increases in cell number, such as sonic hedgehog, VEGF, but the ones that were causing this difference in the activated phenotype were TGF-beta and BMP4. And these are both part of the TGF-beta superfamily, but we found that they were activating by distinct pathways by using inhibitors specific for each receptor. We then wanted to know, right, tumor cells express KRAS, activate stellate cells, how do they then feed back to affect tumor cells? We then used a methodology called CTAP, so cell type specific labeling using amino acid precursors. So the way this works is a little bit like SILAC, but here each of the cell types expresses an enzyme that metabolizes one amino acid precursor, one amino acid precursor. So either a lysine racemase, so you can feed with D-lysine, or DDC. And using these, we were then able to co-culture cell lines that had the receptor for TGF-beta knocked out in the tumor cells. So any TGF-beta that we added would act only on the stellate cells. When we do this, we can track what signaling events come from the stellate cells and feed back into the tumor cells. And we found that when we did this in the tumor cells specifically, we saw increases in things like mitosis, cell cycle, transcription, but also metabolic changes, such as proteins in the mitochondria, proteins associated with ATP synthase, and the TCA cycle. And when we did follow-up, we could see, for example, that we saw increased proliferation with conditioned media from activated stellate cells versus tumor cells. And we also saw a drop in total ATP, which fits with this shift and drop in ATP synthesis that was predicted in the Go annotations. And so in these types of experiments, we're able to detect and dissect the reciprocal tumor stroma signaling networks between two specific cell types. And use things like ontological mining to identify novel and confirmable phenotypes. And using this, we were able to specifically pick apart what was happening with the TGF-beta signaling axis. Since I joined AZ, my focus has become much more within imaging. So in my first role in AstraZeneca, I was in the high content biology group. So we did a lot of multi-parametric image analysis, so looking at DNA damage response. We had a multi-endpoint gene toxicity assay, and we had a big focus on trying to expand image analysis for all, so using easy-to-use platforms such as Columbus. And after that point, I then moved into the imaging and data analytics group, which for me was very exciting. So I now work in the interface between mass spectrometry and multi-parametric image and data analysis, and trying to drive the integration of data from different platforms to use to understand drug delivery, drug metabolism, response, and resistance. So one of my first projects was trying to pull as much information out of a simple stain, such as Hirsched, to increase the amount of data that we could get from our high throughput screens. So normally Hirsched is just used to identify cells and stain the nuclei, but there's an awful lot more information that you can get out of that by employing more advanced image analysis techniques. You can see, for example, micronuclei, which is where a partial or whole chromosome has been lost from the main nucleus. You can track which cells are in mitosis, which cells are in apoptosis and undergoing nuclear fragmentation. You can see which cells have multinucleation. And so using these different techniques, you can understand more about the effect of compounds, particularly in the DNA damage space, or looking at those that cause gene toxicity. So using these types of, one that I should also, you can also look at the cell cycles. The amount of DNA is proportional to where they are in the cell cycle. This is used a lot in flow cytometry, but we wanted to apply this and develop techniques to extract this data from images as well as just from single cell flow cytometry. And we've applied this to multiple different projects in oncology. This allows us to get various endpoints by cell cycle phase to understand if compounds are causing a block in a cell cycle phase, and do this in a dose response and high throughput manner. So we can do this in 384 well plates, and we've applied this to lots and lots of projects now. So I've just got a few here as examples. But this allowed us to go from a point where people were doing 1 or 2 doses, for example, and looking in a very low throughput manner at one endpoint at a time to getting all of this rich information simultaneously. We also can do this to look at genotoxicity. So a lot of compounds we want to check before we go proceed to the clinic, for example, that they aren't causing unwanted DNA damage. And so we use these similar types of techniques to understand, so go from these images where previously they were manually scored for whether they were causing DNA damage to a point where we could make this much higher throughput and faster. And we initially did this using specific markers of different proteins within cells. But what we have seen is we can get all of this information again just from looking at the DNA stain. Because ultimately you're looking at damage to the DNA. And so there is sufficient information within that DNA stain to allow us to pull that information out. Right. So those were sort of my first two roles. So what I'll spend the rest of the talk talking about is what I am doing at the moment. So I work within the imaging and data analytics team, and particularly looking at how we can use mass spectrometry imaging and combine it with other histology approaches to really pull as much information directly from tissue as possible. So for those of you who might not be familiar with mass spectrometry imaging, I've just got this slide just to walk you through how it works. So we start with a fresh frozen tissue section. We then have an ionization source that rasterizes across the tissue. And then for every pixel, you have ions that then go into a mass spectrometer, and you generate a mass spectrum per pixel. You can then pull out specific peaks from within this and build up an image across your whole tissue for different ones and generate multi-channel images from those. There are lots of different types of mass spectrometry imaging, so they have different ionization techniques. So matrix-assisted laser desorption and ionization, and desorption electrospray ionization as two of the main ones. But all of them, the aim is to generate ions that you then put into your mass spectrometer. And then you have different types of mass spectrometer, such as TOF or Orbitrap. But they all vary in their spatial resolutions, that's the distance between two adjacent pixels, and their spectral resolution, so the ability to distinguish two close M/Z values. So we use MSI a lot at AstraZeneca. It's really great for doing things like imaging drug and metabolite distributions in tissues. So we use a lot for PK analysis, say where our drug is getting into the tissue and how much is getting in there. Getting on tissue quantification, so we can get directly quantify drug distributions from tissues. We use it a lot to look at target engagement and efficacy studies, but the things that it is less good at, we can't use it on FFPE tissues. We can't distinguish free drug from total drug. We can't get down to the subcellular level. And in particular, we can't look at large molecules. So we can't look at proteins, for example. And so we needed to find something that would allow us and complement this type of information. And this is particularly important in tissues, as I'm sure you'll know, because you'll have many, many different cell types, all of which are interacting in different ways. And each of those cell types, you need many different markers to identify what each one of those is. And so you have this sort of multi-surface microenvironment of different cells interacting. And so in addition to this label-free mass spec imaging that we can get endogenous metabolites, drugs, and different biomarkers, we also use label-based mass spectrometry imaging, in particular imaging mass cytometry. So this is basically a type of hyperplexed immunohistochemistry technique where your antibodies, rather having fluorophores or chromogens, you have heavy metals. And works in a very similar way to our other mass spec imaging techniques, where you rasterize across a tissue and per pixel get a spectrum, but this time of the metals contained within there. And in this case, you can build up images of where those metals are in the same way as you would visualize your chromogens or fluorophores to get all of that information of the protein localization simultaneously. So we use this in lots of different areas. So as I've said, we quite often use it to look at drug disposition. So we can look at where the drug is and whether it's been metabolized into its inactive or inactive metabolites, and overlay this with things like our H&E information to understand where the drug is getting and where it's going to be active. We use it quite frequently in efficacy studies. So we use it to look at endogenous biomarkers, such as endogenous metabolites like aspartate, glutamine, glutamate. We use it to look at safety. For example, if we have a drug that's crystallizing, we can see where that is and quantify it. We also use it to look at model characterization. And again, this tends to be looking at some of those endogenous metabolites. And I'll just walk you through a few examples of projects where we've used these different techniques in combination. One of these is looking at drug delivery metabolism. So this was, again, in pancreatic ductal adenocarcinoma, but looking at gemcitabine. And so we're looking at gemcitabine delivery and its metabolism into its inactive and active metabolites. So in these images here, you can see we have gemcitabine, the parent compound, its inactive metabolite DFDU and its active metabolite DFDCTP. And what's really striking in these samples is that you can see the parent compound and the inactive and active metabolites all have very distinct localizations. And so it's not as simple as you deliver the drug and where it gets to, it's active. Here you have it being metabolized into an inactive form and into the active form. And when we start looking at some of these endogenous metabolites at the same time, in the same tissues, you can see, for example, heme. So this is a marker of where we have a lot of blood. We can see that the parent compound is high where we have high amounts of heme. And again, if we combine this back with the H&E information, and we say, look, in this region where we seem to have particularly high levels, we can see that this tissue is actually very hemorrhagic and necrotic. And so it's not that we have high levels of delivery, what we have is high levels of hemorrhaging, and so pooling of the drug in these hemorrhagic areas of tissue. However, when we want to look at the active metabolite, we can see we have very little in these dead necrotic regions, but in the adjacent regions, we have much higher levels. And when we look at endogenous small molecules such as ATP, ADP, and AMP ratios, you can see that it's much higher and correlates with where we have higher levels of these ribonucleotides, which would fit with the fact that the active metabolite is formed via ribonucleotide biosynthesis pathway. So it's a CTP analog. And so it needs to be in its active form, it needs to be phosphorylated via this pathway. So where we have higher amounts of this pathway, we have higher amounts of the active metabolite. Next, we wanted to know, does this then correlate with higher levels of its activity? And so we used the imaging mass cytometry. So the relative resolutions between the two techniques are very different. So in MSI, our pixel size is usually around about 50 microns. And you can see these as these large yellow squares of the active metabolite. But you can see where we have the active metabolite, we also have higher levels of gamma H2AX, which you can see in green here. So where we have higher levels of the active metabolite, we are seeing higher levels of DNA damage. And in this way, by bringing these different types of information together, we can get a really good understanding of how these drugs are acting. Another study is then looking at drug response and resistance. So in this study, we had a combination of a PI3 kinase and an mTOR inhibitor. And these we could see have their distinct effects on signaling. But what was particularly interesting is their effects on the metabolism in the tumor. So this is the vehicle, and we can see again, looking at metabolites like ATP and ADP, is that when we hit with the PI3 kinase and mTOR inhibitor, we see a significant reduction in the overall levels of ATP and ADP in the tissue. But what was particularly interesting in this study is that we saw these small regions within the tumors where we didn't see a very big reduction. And likewise with other pathways such as the glucose phosphorylation, we can see a buildup of these glucose glycolytic intermediates within the tissue that, again, we don't see within these specific regions. And so we used these different techniques such as IMC again to pull out the signaling within these regions. And you can see all of these pathways, when we're not inhibiting, are very high, but when we inhibit, we specifically see reductions in these across most of the tissues. But again, not within these specific regions, we still see more of, for example, phospho-AKT. And so this was very interesting, but we wanted to know what were the cell types and phenotypes that were responsible for this apparent resistance within the tissues. So we used H&E, for example, but we couldn't see any significant features, at least by eye, that pulled out different regions that correlated with the MSI. But when we used the IMC, we could begin to see much more differences within these regions compared to the rest of the tumor. For example, we could see that these regions were in fact more proliferative, that we had higher levels of the signaling pathways. We also had differences in the amount of vascularization and differences in the relative epithelial to mesenchymal transition within those areas. And so this is an ongoing study where we're trying to understand why these parts of the tumor are acting very differently. And I hope this sort of brings the power to the sort of power of hyperplex imaging. So at least traditionally, when we looked at tissues, we would look at different markers individually. And so you can quantify and pull out each marker in each one by itself. But what these new technologies are allowing us to do is look at all of these simultaneously and get a much better idea of tissue biology. It's just an example from the kidney using IMC. And so what can we then do with these? Well, we can then look in our projects and use all of these different markers to, for one, segment the tissue into the different regions. So we can say the medulla versus the cortex versus the glomeruli within the cortex. And we can also start to look at different markers within there. And one particular area that's very useful, having many markers, is to look at immune deconvolution, because there are many different types of immune cells and you usually need multiple different markers to define them. So this is an example of a project we had in chronic kidney disease, where we applied this panel and were able to understand what our drug was doing in chronic kidney disease. We could segment out the tissue, but then we could specifically pull out different changes that we were seeing within the immune populations. So for example, we could see we had a significant drop in neutrophil infiltration within the kidney. And we could also see that we had a shift in macrophage polarization. So we didn't necessarily have a change in the total number of macrophages, but by looking at the markers of macrophage polarization, we could see that we had an increase in anti-inflammatory macrophages with the drug versus pro-inflammatory macrophages that were higher without treatment. And obviously, a lot of what I've shown you so far, we have been using more targeted type analysis. So we can use various machine learning techniques to segment tissue into different areas, tumor versus stroma, for example. And then we can go in and manually threshold to identify different cell types, combine these into multi-marker phenotypes, for example, to define neutrophils or to define pro or anti-inflammatory macrophages. And we can look at individual spatial relationships of these. But as you can imagine, we have, for example, with IMC, we can look at over 35 markers simultaneously. And doing all of this individually and looking at the spatial relationships of each of the different cell types we can define within that is very time consuming and slow, and we're not really pulling the most that we can out of this data. And so we're moving more to an untargeted type of analysis and phenotype either at the pixel level or at the cell level, and using things like graph theory to understand the interactions between different cell types, and then using data integration approaches to pull the data sets from H&E, from multiplex analysis to the mass spec imaging for endogenous metabolites and drugs, and pull this information together to understand how our drugs are working, what is happening within our model systems, to really get a better understanding of what is happening within that complex tissue biology. And this has really been enabled by the sort of technological advancements we've had in terms of the imaging technologies, but also the advancements we've had in data analysis, which I think you've heard a lot about today. And so we can now get that systems level analysis rather than looking at each marker individually. And we can get that directly from tissue. We no longer have to do the sorts of things that I did during my PhD and look at one cell type co-cultured with another. We can look at how these cells are interacting in situ in the tissue directly. And we can move away from looking at one marker versus another and get more unbiased, quantitative, multimodal insight into what is happening in the tissue. At least at AstraZeneca, the big change that's helped us is being able to integrate the imaging scientists with those data scientists to define the right questions, the right use cases, and to have that interaction to influence experimental design and maximize the advantage that we can get from both these imaging technologies and also the analysis technologies. And with that, I just want to thank everyone who I've worked with, both from my old team at AstraZeneca, Klaus's team, and also the dynamical cell systems team that I did the cell imaging with, and also the oncogene team that I worked in as well as my team in IDA, the different supporting teams. So the chronic kidney disease team, whose work I presented here, the antibody drug and protein engineering team, the safety sciences team, the discovery sciences team, oncology, as well as collaborators outside of AstraZeneca, including at National Physics Laboratory, ICL, the Beats Institute, and the CRUK CI. And in particular, I want to thank the CRUK Grand Challenge team, who we've been working with closely to develop ways to integrate these different types of imaging data. And thank you for listening.
Thank you very much, Stephanie. Great talk. Okay, so now we'll open for questions. Anyone that has a question, just unmute and speak up. Okay. Yeah, Michael.
Hi, thank you. Thank you for your talk. I have a question more on the career side. I wonder, how do you think working in a big company, and I mentioned AstraZeneca is one of the bigger ones, compares to the academic setting where you usually have one group? Is it much more interactive, or do you actually end up working in the end also in some sort of group that resembles some sort of a research group in an academic?
I would say it's much more interactive, and I think you can see from just some of the teams I've pulled out here from the presentation, but the way that we tend to work is we will work both within our groups, but with different supporting groups, including sort of quantitative biology, different imaging scientists, but all of us will work together to also deliver solutions and answers to projects. So those will be drug projects that will sit within our therapeutic areas. And so you'll be working with multiple teams, both from an imaging and data sciences side, but also working with different teams who have that biological specificity. So one of these would be one chronic kidney disease team, but there'll be multiple within the chronic kidney disease area.
And is this and also in multiple projects across different teams, or are you usually having one main project and then kind of getting in the people you need to?
Multiple projects.
Okay.
Yeah, so we always tend to work on, so especially within our team, because we are sort of experts in the imaging and data sciences side and have sort of specific technologies that don't exist in the rest of the company, we will take problems or challenges from different areas and find solutions and bring new insight to them.
Okay, thank you.
It's okay.
Stephanie, I also have a question, kind of a bit more general with the data that you present. You seem to have kind of endless options for analysis, and with so much data and different markers. So you can ask many different questions, and I guess, so my question is, do you usually have with the collaborators a very specific question that you want to ask to this data, to this type of images, or you also do some, data analysis, unbiased data analysis to generate some hypotheses? How do you usually work?
So it'll tend to be a combination of the two. So a project team will tend to come to us with a specific question that they want to answer. And so we will do targeted analysis typically to deliver the answers to that one question. But a lot of what we try to do is then take the data that we've generated in that project, so that most of the techniques we used are untargeted techniques. So the MSI data, you always get that full spectrum per pixel. And so we tend to use those data sets to work with the data scientists to see if we can identify any new hypotheses.
Okay.
So in a recent project, they had specific answers, specific questions. We delivered answers to those, but then we also said, hey, by the way, we also found these things. And by working across several projects, we tend to be able to pool data from different model systems, different perturbations, to be able to drive that greater insight that the one project might not be able to find by itself.
Mm-hmm. More questions?
Thank you, Stephanie, for a great talk. I really enjoy it. Fantastic.